Have you ever wondered how scientists figure out the makeup of different substances? Well, they use a tool called a spectrophotometer. The spectrophotometer, an unassuming piece of equipment, is a powerhouse in the realm of scientific research. It's a device that measures how much a chemical substance absorbs light by assessing the intensity of light as a beam passes through a sample solution. This relatively simple concept forms the basis for a multitude of scientific pursuits. In the field of chemistry, spectrophotometers are the unsung heroes. They help chemists identify and quantify substances in a solution, providing crucial data that shapes our understanding of chemical reactions. They allow us to understand the properties of atoms and molecules, and even help us predict how they might behave under different conditions. But the spectrophotometer's influence isn't confined to chemistry. In physics, it plays a crucial role in the study of light and its properties. It allows physicists to delve into the mysteries of light absorption, reflection, and transmission, broadening our understanding of the universe. Biology, too, reaps the benefits of this powerful tool. In labs across the world, spectrophotometers are used to study biological samples, helping researchers uncover the secrets of life. They can be used to analyze DNA, proteins, and other biological molecules, aiding in the development of new medical treatments and therapies. And it doesn't stop there. The spectrophotometer is also a crucial tool in clinical applications and environmental studies. Clinicians use it to analyze blood and other biological samples, aiding in disease diagnosis and treatment. Environmental scientists use it to monitor pollution levels, helping us understand and mitigate our impact on the planet. All these applications and more are made possible thanks to the humble spectrophotometer. Its ability to measure light absorption has proven invaluable across multiple scientific disciplines, driving research and innovation in countless fields. Without the spectrophotometer, many of the discoveries and advancements in science we see today would not be possible. Now that we know the importance of a spectrophotometer, let's delve into how this incredible tool works. Imagine a beam of light. Think about a rainbow, the way it splits into a spectrum of colors when it passes through a prism. The spectrophotometer operates on a similar principle. It uses a device called a monochromator, essentially a scientific prism, to split a beam of light into its spectrum of colors or wavelengths. Once the light is split, the spectrophotometer measures the intensity of this light. This is where the magic happens. But how does it do it? It's all about detectors. These detectors measure the intensity of light before and after it passes through a solution. They are like the eyes of the spectrophotometer, keenly observing and noting every change in the light's intensity. Now you might be wondering, why is it so important to measure the intensity of light? Well, let's think about a cup of tea. You can tell how strong the tea is by looking at its color, right? The darker the color, the stronger the tea. This is because the tea leaves in the water absorb light, and the amount of light absorbed can tell us the concentration of the tea. It's the same with the spectrophotometer. By measuring the intensity of light before and after it passes through a solution, it can determine the concentration of the substance in the solution. It's like the spectrophotometer is tasting the solution with light, but it doesn't stop there. The spectrophotometer doesn't just measure the concentration of a substance. With different wavelengths of light, it can even identify what the substance is. It's like having a superpower, being able to see the unseen, to decode the mysteries hidden in the light. So, in essence, the spectrophotometer is a tool that measures light to reveal the secrets of the universe. It's a beacon of knowledge, shining a light on the mysteries of our world, one wavelength at a time. It's time to go a bit deeper and understand the nuts and bolts of a spectrophotometer. Imagine the spectrophotometer as an orchestra. Each instrument or section has a unique role to play, but they all work together to create a symphony of scientific discovery. Just like that orchestra, a spectrophotometer has 4 major components, each with a specific function that contributes to its overall performance. First up, we have the light source. This is the drum section of our orchestra, setting the rhythm and providing the energy. The light source emits a beam of light that starts the whole process. It's like the spark that sets off a firework, illuminating the path to discovery. Next, we have the monochromator, the conductor of our orchestra. It's the component that selects a specific wavelength of light from the broad spectrum produced by the light source. Think of it as a filter. It only allows a particular color or wavelength of light to pass through while blocking the others. It's a bit like choosing only one instrument's sound out of the entire orchestra. Then, we have the sample holder. This is the stage where the performance happens. It holds the sample, the subject of our investigation, and allows it to interact with the light. It's like the spotlight on the lead performer, focusing all attention and energy on them. Finally, there's the detector. This is the audience in our orchestra analogy, listening, observing, and recording the performance. The detector measures the amount of light that passes through the sample. It's the critical component that gives us the data we need. It's like an art critic, meticulously noting down every detail of the performance. The light from the source travels through the monochromator, which selects a specific wavelength. That light then hits the sample in the holder. Depending on the sample's nature, it will absorb some of this light. The remaining light then reaches the detector, which measures the intensity of this transmitted light. From this measurement, we can determine how much light the sample absorbed. This data can tell us a lot about the sample, like its concentration, composition, and even its structure. It's like understanding the story behind a song by listening to its lyrics and melody. Remember, all these components work together in harmony. The light source provides the energy, the monochromator selects the specific wavelength, the sample holder presents the sample, and the detector measures and records the data. Each part is crucial, and without one, the whole symphony falls apart. So, that's the spectrophotometer for you. Not so intimidating when you break it down, right? It's a marvel of scientific engineering, each component working together to unlock the mysteries of the world around us. From the light source to the detector, every part of the spectrophotometer plays a critical role in helping scientists unlock the mysteries of the world. We've discovered quite a lot about the spectrophotometer today, haven't we? This little device, often overlooked, is a titan in the science world. Its role in scientific research is undeniable, and it's time we give it the recognition it deserves. We started our journey by understanding the importance of the spectrophotometer in scientific research. It's not just a fancy piece of equipment sitting on a lab bench, but a critical tool that enables scientists to measure and analyze the properties of light over a specific portion of the electromagnetic spectrum. Thanks to this device, researchers can understand the interaction between matter and radiated energy, a fundamental aspect of scientific studies that span across numerous fields, from biology and physics to chemistry and environmental science. Then we demystified the spectrophotometer, peering into its working principles. At its core, a spectrophotometer operates on the principle of light absorption. When a beam of light hits a substance, some of it gets absorbed while the rest passes through. By analyzing the intensity of the transmitted light, the spectrophotometer can calculate the amount of light absorbed by the substance, leading to a wealth of information about its properties. We also took a closer look at the key components that make a spectrophotometer work. The light source, which can be a bulb or a laser, provides the initial light beam. The monochromator, often a prism or a grating, separates this light into its different wavelengths. The sample holder, where the substance to be analyzed is placed, is where the magic happens. This interaction between the light and the substance provides the crucial data that scientists analyze. Finally, the detector measures the intensity of the transmitted light, and the readout device, such as a computer, presents this information in a form we can understand. But a spectrophotometer isn't just a tool for scientists to play with in the lab. Its practical applications are vast and varied, from quality control in the food and beverage industry to drug discovery in pharmaceuticals. From detecting pollutants in water to diagnosing diseases in medicine, the spectrophotometer is a versatile player in our everyday lives. So the next time you bite into a perfectly ripe apple, drink a glass of impeccably balanced wine, or take a pill for your headache, remember the role of the spectrophotometer. And when you read about a new scientific breakthrough, think of this humble device. It's likely that behind the scenes a spectrophotometer was hard at work, providing the data that made the discovery possible. Next time you hear about a scientific breakthrough, Remember the humble spectrophotometer, the unsung hero of the science world, which plays a crucial role in making it happen.